Okay, welcome to Chapter 3, the molecules of cells. All right, now in the previous chapter, we went over molecules, we went over compounds, the elements, bonds. Now we're going to go over something called biological macromolecules. Okay, does anyone know what macro means? So think about that term quickly. Macro means large, and molecules means molecules. So macromolecules are large molecules. Now, these that we're going to talk about today are essential for life. What does essential mean? Essential means that you cannot live without it. They're essential for life. Okay, there's 4 biological macromolecules that are essential for life. Carbohydrates, proteins, lipids, and something called nucleic acids. All right, now all of these are organic compounds, and these are the biological macromolecules of life. Now let's talk about organic and what exactly that means. Now the term organic is in reference to an organic compound that is made primarily out of the element carbon. Okay, now the bottom line is that it has to be made of at least 2 or more carbons. Therefore, a compound or a molecule that has less than 2 carbons, for example, let's see, I'll give you a couple of examples. These are inorganic, not organic. Inorganic would be CH4 or CO2. Those are inorganic. Okay, therefore, in other words, an organic compound is made mostly out of carbon, and it has to have at least 2 or more. And this is an incredibly significant part of life. In fact, there's an entire field of science dedicated to studying organic compounds that is called organic chemistry. Okay, now everybody knows carbon because we worked with carbon in the last chapter. Therefore, an organic compound has to have a minimum of 2 carbons. This is the smallest organic compound. This is called ethane. See, it has 1, 2. And look what else it has. Hydrogen, hydrogen, hydrogen. Okay, these are together are referred to as hydrocarbons for the simple fact that it's made primarily out of hydrogen and carbon. And notice they get bigger and bigger and more complex and even start to make rings and shapes. Now, this isn't an organic chemistry class, but the point is we need to know the fundamentals to understand everything that we're working with. Now, all of these macromolecules that we're going to learn today have a hydrocarbon backbone, and they are the compounds that make up life, make up living organisms, including the human body. All right, now, these compounds can become more complex by adding a functional group to them. And we're just going to go over a couple of the functional groups. So basically, let's say we're talking about propane. If we replace this H with a functional group, one of these, it will completely change the composition of it. Okay, so the first one is an OH group. Okay, and adding an OH to the elements makes it an alcohol. Okay, the next one's a C double bond O. That's a carbonyl group, and it can be shaped like this, or it could be located at the end. That's an aldehyde, or in the center, and that would be called a ketone. Okay, this is a COOH. That's a carboxyl group. Something that has an NH2 or an NH3, like this one, is an amino group. Okay, anything that has a PO3 or a PO4 is a phosphate group. This one's pretty important. We're going to see it quite a bit. And a CH3 group like this is called a methyl group. All right, so adding those functional groups to an element, a compound, can completely change its function. Okay, now we are essentially building our way up to discussing each and every macromolecule, the 4 that were introduced. Now, how do you make a macromolecule? Now, the system, our body, our cells, knows how to do this. And there's one way, and one way only, to make a macromolecule. And it is through a chemical or biochemical reaction called dehydration synthesis. Okay, now we are using, or they are using, a term called polymers. Okay, they're going to use terms like polymer and monomer to indicate a non-specific subunit of the macromolecule. So this is a non-specific macromolecule. Okay, one of the compounds, one of the units that are going to be added to the macromolecule is referred to as a monomer. Mono means one. Okay, a combination of them, such as this right here, there's 1, 2, 3, that is called a polymer. Okay, now we can make it even longer and longer and longer through one reaction, dehydration synthesis. Now it's called that because if you notice, there's an OH group on this polymer and there's a hydrogen on this monomer, and together that's an H2O. And what happens is a water molecule is removed in this biochem reaction, producing a connection right there. Okay, and a new bond is formed to make a longer polymer by removing one water molecule, thus the term dehydration synthesis. Dehydration obviously is the loss of water. Synthesis, now this is important. All of it's important, but I like to teach my classes little terms that you're going to see again and again and again. And they might be used in different ways, and they might emphasize a different segment or a different chapter or a different topic, but the facts remain the same. Synthesis means to build. Synthesizing something is to make, to build. Okay, so dehydration synthesis means to build by dehydrating. And that's exactly what happened. We just built, we just attached a monomer. We just built a polymer. Well, we extended a polymer. All right, so you should remember this word. You'll see it again in other chapters. What about breaking down a macromolecule? All right, for every reaction, there's an equal and opposite reaction, right? So if we can build macromolecules, therefore, we should be able to break down macromolecules in the exact reverse order. Okay, so we built them by removing one water molecule. Therefore, we can break them down by adding a water molecule. And that's exactly what's happening here. So we have 1, 2, 3, 4 units in our polymer, and we're going to add a water molecule and it will immediately break the bond. This is called hydrolysis. Okay, all polymers are broken down by hydrolysis, one monomer at a time. Now I want you to remember this as well. the term. So you're going to see a lot of science terms, and sometimes they can be a little bit intimidating. Now, you don't want to be intimidated by it at all, and there's no reason to be, because it's not that scary. Hydro, guess what that means? Water. Lysis, and this is a good one to remember. Lysis means to break, not hard. You put it together, to break with water, and that's exactly what's happening here. Hydrolysis or hydrolysis, but you want to remember those, hydro and lysis, and of course, synthesis. These kind of terms will help you kind of smooth sail a little bit more. All right, so let's go on. All right, so we're going to go through one carbohydrate at a time. I'm sorry, not carbohydrate, one polymer at a time, and break them down and learn everything about them. All right, so the first polymer, or in other words, macromolecule, is the carbohydrate category. There's different types of carbohydrates. Now, we have something called monosaccharides. Okay, now think of what mono means. Mono means one. I'm going to make another box over here for note taking. Okay, mono means one. What about saccharide? What the heck does that mean? Saccharide means sugar. Okay, put those two together. Monosaccharide means one sugar. Okay, great. Easy. What about disaccharide? Di means two. That means two sugars. Okay, anything bigger than that? Poly. Poly means many. That's what poly means. Okay, polysaccharides just means many sugars, more than two, more than, it could be 3, it could be 3,000 or 300,000. All right, so it's just more than 2. Okay, so let's go ahead and take a look at a couple of the more significant monosaccharides. Okay, these are referred to as simple sugars. They are composed of a single sugar monomer, one sugar unit, monomer. Okay, now glucose is something that you're going to see again and again and again, and the reason why is because our body, actually all living organisms, take glucose and turn it into energy. Well, we can break it down into the energy. Now, this is very important. You're going to see this again and again. Glucose is equal to energy. Let's go ahead and write that. That is something you want to keep at the forefront of your mind throughout this course. All right, glucose is equal to energy. And we will see exactly why in later chapters, but that one's incredibly important. Okay, now notice this is a 6-carbon sugar because we can count them up. There's carbon number 1, 2, 3, 4, 5, and 6. Okay, fructose, on the other hand, is a 5-carbon sugar and makes a pentagon. All right. If we pull these apart and I break down that whole, this whole hexagon, we'll see what the backbone is. And guess what? It's a hydrocarbon backbone. Okay, there's glucose. And look at that. It's nothing but carbons and hydrogens and oxygens. And this goes back to the first thing we discussed in this chapter, which is all organic compounds are made of a hydrocarbon skeleton. Okay, now what we're looking at here is glucose and fructose it is unwound. Okay, so now the next thing we're looking at is, of course, honey and bees in their little hive. And why would that be there? Because that is where we get our sugar. That's part of where we get our sugar. That's a good sugar resource. So they toss it in there for good measure. Okay, what about a disaccharide? Well, first of all, the cells have to construct a disaccharide. Plant cells can do it, our cells can do it, bacteria cells can do it, so it's not too hard. All they have to do is the dehydration synthesis reaction. Now, actually, this is kind of significant. Sometimes in the book, you'll see them flip-flop the term dehydration synthesis and write dehydration reaction. I want you to know both terms mean the same exact thing. All right, so here we are connecting one glucose with another. There's only one way to do that, and that is through dehydration synthesis, removing one water molecule, and they will immediately connect. And this makes a disaccharide called maltose, 2 sugars. Maltose is pretty interesting. Maltose is used to make beer, malt liquor, malted candy, and remember, it's made through dehydration synthesis. Okay, so these are some of the things that we have in our life. Okay, what about this one? This one's lactose. Lactose is a sugar. It's actually a disaccharide specifically that is found in milk. How do you build lactose? Okay, there's only one way to build, and that's dehydration reaction. Okay, so this is lactose. It's glucose plus a sugar called galactose. And in people that are lactose intolerant, they lack the enzyme, okay, that can cut through this. Now, and they become lactose intolerant, so that if they drink milk or eat something that is dairy, they get upset stomach and things like that because they cannot break this down into 2 monosaccharides. All right. Okay, our next topic is polysaccharides. Now remember, poly means many, and saccharide means, of course, sugar. Okay, now this is a macromolecule, and there are several different types that we're going to talk about today, but the important thing to understand is that, in fact, this is not a monosaccharide nor a disaccharide. In fact, some of these have hundreds of thousands of monosaccharides linked together in a chain, which we refer to as a polysaccharide. Now, how do you build a polysaccharide? Which reaction is used? Okay, dehydration synthesis, also called dehydration reaction. There are 3 common polysaccharides. All right, the 3 most common polysaccharides are starch, glycogen, and cellulose. Now, what we're looking at here, I'm going to zoom in on it. Okay, this is a picture of a person that is collecting or picking up potatoes. And see these arrows coming out of the potato. All right, the whole point of that is to show you that this is where we can find starch. And if we were to put that under the microscope, zoom in a little more, and we were to analyze, we'll see these little starch granules under the microscope inside of, this is actually a plant cell around here. This is, all right, so these are little starch granules, these little like bubbly looking things, okay? And inside of there is starch, and this is what starch looks like on a molecular level. See over here how there are these individual units. These are glucose subunits. In other words, glucose monomers. And they go on and on and on and on. It goes on and on and on. I mean, we're talking hundreds of thousands of glucose monomers. Okay, that's glucose. And that's starch. Now, starch is found inside of plants. Okay. Next, glycogen. Now, see this person's arm and they pulled out a muscle and you see an arrow coming from the muscle. This is in reference to the analysis of what's inside of human muscle tissue and actually all animal muscle tissue. Okay, this is a muscle cell in this figure, and inside what we have is glycogen. Glycogen is this triple chain. See, there's 1, 2, 3, this triple chain of glucose monomers. And it is pretty long, and you can find it in every single human muscle and the human liver. And actually all animals store glucose in this manner. Okay, next, cellulose. So here we have a leaf or some kind of plant, and the arrow is zoomed out on that, and then it breaks down cellulose for you. And actually, if you look at this picture, this part of the picture here, it looks sort of like a cable cord or that's been cut, some kind of wire that's been cut. And if you look at the breakdown, all of these are glucose subunits, right? That shape, that hexagon, that is our glucose. Okay, and then there's 3. There's 1, 2, 3 chains of it. Okay, and then each chain is connected by hydrogen bonds. And these, actually sugar in that row is connected by a hydrogen bond. This means that this thing is rock solid tight. That means, in other words, we cannot even break down cellulose. All right, so let's talk a little bit more about these 3. Now, earlier we said that glucose was sugar. I'm sorry, yes, sugar, but specifically glucose is equivalent to energy, and that is still true. Now, so in short, starch is how plants store energy. They take up glucose and they can attach it to the chain of starch, or start attaching, 2 monosaccharides to make a disaccharide, and then continue to add glucose monomers to make their polysaccharide of starch. But this only happens in plants. That's all. Now, we get the energy from our diet. We get this energy that plants store away. Okay, we pick it up in the form of starch by eating potatoes, grains, which is everything from wheat, corn, rice. Of course, there's pastas, things like that. That's where we get our glucose storage. Okay, glycogen. Glycogen is how animals and humans, we are animals as well. We are in the animal kingdom. So animals and humans, that term is biologically synonymous. All right, now either way, glycogen is how we store our energy. This is energy storage in humans. Okay, so remember, it's here in this slide, it says glucose is stored in animals as glycogen. Yes. Now, what was the note that we took earlier? Okay, we said that glucose is equal to energy. So in other words, glycogen is animal storage of energy. Okay. There's 2 primary locations for glycogen. Okay, you can find it in muscles and the liver. That's where we store our energy. What about cellulose? Okay, cellulose was that triple chain held together with hydrogen bonds, and in fact, this component, that's found in plants, it's also found in wood. It makes up the plant cell wall. That's what it does. It's also found in wood. Now, animals like us, we cannot break down cellulose. We lack the enzyme required to break down cellulose. Therefore, when we eat cellulose in our diet, it comes out as fiber. Okay, so what's interesting about this is termites, little house termites. They can destroy an entire house, right? Especially the houses in the United States, which are primarily made of wood. Okay, and how do they do that? Because they possess this particular enzyme that can break down cellulose. That's interesting because, of course, we lack that enzyme. Most animals lack that enzyme. Insects and things like that lack it as well, except this one. This is the one that has the enzyme to break down cellulose and, in turn, destroy a person's home. So, okay, now this is a great chart, and I want you to draw it out yourself. Now, you can pause the screen at any time and copy this stuff down. And you would normally take these notes inside of my classroom and they would be written on the board. But I've made this chart for you guys because we're online this semester. Well, this whole year, really. All right, so what you want to do is be able to analyze any chart that I give you or that you see. So, of course, look at the very top. It says macromolecules. So, of course, this whole column is a list of the macromolecules. Carbohydrates, proteins, lipids, nucleic acids. Okay, then it has the monomer subunit for each, and then it has the polymer subunit for each, or I'm sorry, not subunit, the polymers in general. Okay, so all we've talked about so far is carbohydrates. So you want to fill that in, carbs. The monomer subunit is monosaccharides, and it could be glucose. That's the main one we've discussed. But it also could be fructose. But the fact is, it's a monosaccharide. Now, these polysaccharides that we want to discuss or that we have discussed, starch, glycogen, cellulose. I haven't actually talked to you guys about chitin. I'll quickly discuss it. Okay, chitin. Okay, it makes up the exoskeleton of insects. Okay, so bugs, insects, they have an external exoskeleton and it's made out of a polysaccharide called chitin. All right, let's go to the next macromolecule. Okay, lipids. Now, lipids are interesting because they are technically not a macromolecule, but they're not a huge macromolecule. They're not a polymer, but they still fall under the 4 essential macromolecules, and they do have certain things in common. They are very diverse. I'm going to teach you the diversification, and I'm going to teach you guys the similarities. Now, lipids, one thing they have in common, all of them, no matter how different they are, they're all hydrophobic. Remember, hydrophobic means water-fearing, which means they can't be dissolved in water. The 3 most important types of lipids are fats, phospholipids, and steroids. All right, this is the structure of a typical fat. It has a subunit up here. This is called glycerol. And then it has this big, long, fatty acid tail. And see, it's just nothing but carbons and hydrogens all the way through. This is completely hydrophobic. Now, glycerol plus a fatty acid tail to connect them what's interesting is even though it's not a big, long, polysaccharide like we saw earlier, or a big chain, there's still only one way to build the fat. And you have to use dehydration, synthesis to connect. And then these two will be connected, removing one water molecule. Okay, so that's the typical structure of a fat. Now we also have a triglyceride, or right, this is a triglyceride, and it's called that. It's a fat with 3 fatty acid tails because tri means 3, and it's called the triglyceride because of this molecule glycerol. All right, so there's regular fats, then there's these fats that have a triple fatty acid tail, 1, 2, 3. And not only do we have those two, we have unsaturated and saturated fatty acids. Okay, so this is the difference, and I'm going to go over this quickly because it's important, but it's not a very complex subject. This is the bottom line. Okay, unsaturated fats. We're typically told that they're healthier, and in room temperature they are liquids, like olive oil and oils in general. And the reason why they are liquid at room temperature is because of this. Okay, see, this is the glycerol head and the fatty acid tail. Now they have unsaturated fatty acids have a double bond in the hydrocarbon chain. See this little double bond, double bond, double bond, double bond. Now what happens is when they're put into a group, okay, they are not able to stack together tight. There's space in between because of the kink in the tail due to the double bond, making this liquid at room temperature. Okay, now the next thing is the fatty acid, the saturated fatty acid. This has no double bond, and therefore these molecules can stack together tight. Okay, like in this picture. See how these guys, there's no double bond, there's no space, stack, 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 stack with no problem, therefore solidifying at room temperature. And these are technically, the saturated fats are the unhealthy fats. Okay, so that's the big difference. These guys have no double bond. These guys have a double bond. And then you have this drastic difference. Okay. This is just a quick picture of what we mean by unsaturated and saturated fats. Everyone should kind of know this just from, TV and life. But there we go. See this olive oil and avocado and vegetable oil and nuts, bad fats, or butter and lard and things that are solidified. Next, phospholipids. Now we're going to talk quite a bit about phospholipids in chapter 4 and 5. However, for now, I want you to know these simple facts. Phospholipids are a lipid, and they are similar to fats in the sense that they have a glycerol. There's their glycerol, a fatty acid tail. This one has 2 fatty acid tails. However, what makes them different is they have this entirely polar or hydrophilic head. See this area where it says polar head? I want you to note that. This is hydrophilic, which means what? Hydrophilic means water-loving. Okay, all of this is fatty acid tail is very hydrophobic, very. Okay, this is significant. All right. Now, that's the basic structure of a phospholipid. Now, what do? What's the purpose of them? They are the primary component of your cell membrane, of my cell membranes, of every cell membrane of every organism on the planet is made out of these phospholipids. So see here, this is a cell, and all of this outside layer is the cell membrane made completely out of phospholipids. All right, now we'll go more into the detail of the anatomy of the cell membrane in the next chapter, but for now, you want to know this. Phospholipids make up the cell membrane. Actually, it's not the only thing that makes up the cell membrane though. So it is the primary component. Next, steroids. Alrighty, now steroids are actually something that your body makes and that your body needs, like hormones, and they just fall under this big blanket, umbrella category called steroids. We'll draw a little umbrella. Okay, that's a terrible umbrella. Sorry, guys. You get what I'm saying. All right, so steroids, what falls under here are your hormones. And the reason why is because of their structure. Now, these are absolutely tiny. And what's interesting is every hormone is going to have a different structure, different function. But these hormones that we're talking about today, like the sex hormones, which is estrogen, testosterone, they have a cholesterol-based backbone, which is this. Okay, see this structure here? Let's zoom in on it. If you count this up, this is 1, 2, 3, 4. That's 4 rings. Okay, and in short, these we refer to them as a carbon skeleton bent to form 4 fused rings. Okay, this skeletal backbone is the skeletal backbone of the sex hormones. This picture is showing cholesterol, but estrogen and testosterone share this in common. Therefore, steroids have this standard shape in common, the 4 fused backbones. Now, they might have a different R group over here, or this might be different here, or this might be different here, but the fact is that these 4 fused rings make it a steroid. Okay, so if you're drawing your chart as you're supposed to, now you're going to add in lipids, and the way I've organized it is this. I've made rows, 3 rows, okay, 1 for fats, 1 for phospholipids under polymer, and 1 for steroids. And then I've given the monomer subunit, okay, and for fats it's glycerol plus fatty acid. For phospholipid it's the polar head plus fatty acid tail, and for steroid it's 4 fused rings. Okay, the next topic is proteins. All right, now out of all of our polymers, we have monomer subunits, right? So that's an important factor. And for proteins, the monomer subunit are amino acids. Okay, amino acids are linked together through one reaction, dehydration synthesis, to make our proteins. Okay, however, when 2 amino acids are linked together, they form something called a peptide bond. We have 20 amino acids in nature. That's it. So proteins are a combination of 20 amino acids, or there can be repeats of 1 or 2 or more amino acids, but the fact is that we end up with these long chains and complex 3D structures that make up our proteins, our polypeptides, all of it. Okay, so all proteins are made of amino acids. A chain of amino acids is also called a polypeptide. And why would we call it that? Polypeptide. Well, poly means many, and peptide is in regards to the peptide bond that is formed between the two. Every amino acid has the same structure to an extent. Okay, this is what they have in common. They have a central carbon right here. Okay, carbon can make 4 bonds. 1, 2, 3, 4. Okay, so it always has an H group somewhere, or sorry, not an H group, a hydrogen. And then to the left, there's always an amino group. Remember, amino groups, they always have an NH. It can be an NH2, it can be an NH3, as long as there's an NH something, that is an amino group. And so it shouldn't be surprising as to why we call these amino acids. Okay, because the next group on the right here is a carboxyl group, and that's a C O O H. Okay. Now, okay, so how do we have 20 different amino acids if they all have the same structure? Because of this, because of the R group right here. This R group, there are 20 different R groups. Okay, 20 different R groups that distinguish or make the 20 different amino acids, but everything else is the same. Every amino acid has an amino group, a carboxyl group, a central carbon, and the hydrogen, and then one of the 20 R groups. Okay, very good. Now, proteins are connected or built by dehydration synthesis. It's a bond. There's a bond between 2 amino acids. Okay, and every time there is a bond, there's only one way to connect them. Okay, this is very significant. So I'm going to diagram on here. This is one amino acid, and it's nonspecific. This is a second. There's a plus sign in between, so we want to add them together. And anytime 2 amino acids connect, the carboxyl group of one amino acid connects to the amino group of the other amino acid. Okay, and then of course they connect through dehydration synthesis, also called dehydration reaction. So the water molecule is pulled out. So we're removing an H2O from the 2 amino acids, and they will immediately connect and form a dipeptide bond, or a dipeptide with a peptide bond. Now remember, di means 2. So this is one, this is the other, but they're connected. So if I wanted to add a third, I would do the same thing. It would get connected on this side or this side. All right, now proteins are one of the most significant macromolecules. I mean, they're all very significant, but proteins are very diverse. They control everything, nearly everything in the body's function. Okay, for example, every physical manifestation that you have in your body, your eye color, your hair color, your skin color, all of these things are because of your proteins. Okay, they also make up our antibodies, okay, your immune system, the antibody defenses, our proteins. Our cells have proteins that can signal to one another and communicate, sending important messages to neighboring cells. And all enzymes are proteins, and enzymes they catalyze chemical and biochemical reactions. All right, that's just a couple of examples. Okay, let's go ahead and move on. Now, this is important, and you should know this. There are 4 levels of protein structure: primary, secondary, tertiary, quaternary. Okay, now I just make something a little clear. Primary is 1. Okay, secondary is obviously 2 degrees. Tertiary is the 3rd level. Tertiary means 3. And in fact, I'm going to add this 3D. That's what that actually means. In quaternary is the 4th level. Okay. Now, a protein that has a primary structure is simply this. It's a chain of amino acids. So if you look closely, see these 3 letters? This is representative of, it's an abbreviation of an amino acid. So amino acid, amino acid, amino acid. This whole thing is one long straight chain of amino acids. That is the primary structure of proteins. Secondary structure, they get more complex, okay? That it's not just the primary, chain of amino acids. What happens is the amino acids start to take on a form. Okay, this one right here, this is called an alpha helix. Okay. This, see how this is kind of, it's hard to draw it here, but it's kind of this bendy, kind of looks like a spring. That is what a helix is. So you're going to see this word helix quite a bit, and I really just don't want you guys to freak out when you see a word like that, because this is a helix, this shape. So anytime you see the word helix, that's what we're talking about. It's the shape. This one in particular is called an alpha helix. Now, the other shape that's found in secondary protein structures is a beta pleated sheet. So if you look closely at this is pleated, like pleats inside of a skirt or clothes, right? You see how it folds in and up and in and up? Okay, so that is called the beta pleated sheet. Those are the 2 types of secondary structures. Okay, tertiary structures, the protein takes on a 3-dimensional shape. And of course, it can have both alpha helices, beta pleated sheets. All right, now the quaternary structure is 2 or more proteins that join together to make 1 protein. Okay, that's really the ultimate definition of the 4 levels of protein structure that you need to know for Biology 100 level. Alrighty, so back to the chart. Proteins, the monomer subunit is amino acids. The polymer is technically a polypeptide. However, it is also a protein, and don't forget there's 4 levels of protein structure: primary, secondary, tertiary, quaternary. All right, last, nucleic acids. There are 2 types of nucleic acids, DNA and RNA. DNA is deoxyribonucleic acid. RNA is ribonucleic acid. Now, your entire blueprint for your physical body, for everything that is that makes you you, is encoded in your DNA. So we refer to this as our blueprint because of the fact that architects use blueprints to build buildings and things like that, right? Well, our human blueprint is written in the DNA. So it makes us who we are, and it determines how our body functions, whether there's disease or no disease, etc. This is genetic disease, I mean. All right, now ultimately what's put into our DNA here is information used to make our proteins. Okay, now what are nucleic acids made out of? The monomer subunit is something called nucleotides. Those are the monomers of nucleic acids. Now there's one for DNA and one for RNA, but they're still called nucleotides. The nucleotide structure of DNA is as follows. A 5-carbon sugar called deoxyribose, okay? And it's bonded to a phosphate group right here. And then on the other side, there's a nitrogenous base. Okay, we have 4 bases that can be there. In this picture, we have adenine, and that's fine. But there can also be thymine, guanine, and cytosine. This is for DNA. And no matter what's happening with the DNA, the nucleotide, it can only look this way for DNA. It has to have deoxyribose, the phosphate group, and one of the 4 bases. That's it. Now, each strand of DNA is a polymer of nucleotides, and it's composed of a sugar-phosphate backbone. Sugar-phosphate backbone. Now, why would we call it that? Well, for the simple fact that the backbone is a sugar phosphate, the sugar being deoxyribose in blue there, and phosphate being in yellow. So if we look at this ribbon structure, there's your sugar phosphate base, sugar phosphate, and then the base sticks outwards. Okay, so the backbone is nothing but sugar phosphate, sugar phosphate. All right, so in other words, or simply put, DNA has a sugar phosphate backbone, and then, of course, the nitrogenous bases. Okay, now DNA in its natural form is a double-stranded helix. Now, that's kind of a mouthful, but if you break down the terms, double, what does that mean? 2. Strand is a strand, and helix, remember what I just said about the helical shape? Is the helix. Okay, that's it. So DNA is a double-stranded helix with a sugar phosphate backbone. And guess what else? The nitrogenous bases base pair very specifically. A will only base pair with T and C with G. Only. And they're connected by these dot, 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 dot. What does that mean? Okay, if you don't remember from the previous chapter, those dots are representative of hydrogen bonds. Okay, so that's DNA, the intro to it. RNA. RNA is similar and very different at the same time. Okay, for example, it has a 5-carbon sugar, but this sugar is called ribose rather than deoxyribose. It does, however, have a phosphate group, and that's the same as DNA. And then it does have a base on this side. Okay, but the bases are different to an extent because RNA does not have thymine. Does not have thymine. That T for thymine is replaced with something called uracil. Uracil is only found in RNA only. Okay, so the bases are A, G, C, U. Unlike DNA, that's A, T, C, G. Now, RNA is also single-stranded. And if we compare and contrast the two, pretty straightforward, okay? Let's start with the sugar, deoxyribose. If you check this out, the difference between deoxyribose and ribose is this right here. In RNA ribose, there's an O in oxygen, an OH, and here there's just an H, and that completely changes the disposition of the sugar. Okay, then so the DNA has deoxyribose, phosphate, the bases. Okay, in regards to bases, it's adenine and thymine that bind, and cytosine and guanine that bind. RNA has the sugar ribose, it has phosphate, and it has bases. And in this case, if they were to bind, it would be uracil and adenine, and cytosine and guanine. And a really easy way to remember this, the connections I mean, is G always binds with C, and they kind of look alike, so it's easy to remember. And other than that, it shouldn't be a problem. Same thing over here. G will always connect to C, and they cannot connect into anything else. That's it. Those are the rules. All right, so let's kind of recap and wrap this chapter up. In regards to the 4 macromolecules that are essential for life, that would be our carbs, proteins, lipids, and your nucleic acids. The monomer subunit of carbohydrates, monosaccharides. The polysaccharides that are most common, starch, glycogen, cellulose, chitin. Proteins, the monomer subunit for proteins, amino acids. The polymer is polypeptide. Okay, for lipids, there's 3 primary polymers. Technically, they're not polymers, but you get what I mean. The category of lipids has fats, phospholipids, steroids. If you break down a fat, the monomer subunit is glycerol plus fatty acid. Phospholipid, polar head plus fatty acid. Steroids, 4 fused rings. Okay, DNA and RNA for nucleic acids, that is the polymer. The monomer subunit is a nucleotide, and that term goes for both nucleotides, for the DNA nucleotide and the RNA nucleotide. And of course, how do you build these polymers? Through one reaction and one reaction only, and that's dehydration synthesis reaction. Okay, that is the end of chapter 3.